Jim MacDonald. I'm one of the co-hosts for this workshop, and this is the annotation workshop. So one thing about this workshop is normally this is a 2-hour workshop that Lori and I put on, and we don't have that much time, so we're going to be a little rushed, and we're probably going to skip over some of these sections. But if you have any questions, put them in the chat or the poll in Pathable, and we'll try to get to those if we can. So this is just the beginning stuff, saying what you're supposed to know. These are the packages we're using. The idea here is just to teach everybody how to use the annotation packages that we produce for the Bioconductor. So what do we mean by annotation? In general, at least back in the day when people were doing microarrays, what people thought of as annotation is the sort of thing where you've got an ID, you say it's your microarray ID for a certain thing, and you want to say, well, what's the gene that that thing is measuring, or maybe what's the gene symbol, or you might want to get the KEGG protein ID, something like that. But in fact, it can be any of those things. So you could have a set of data that you got that you just have gene symbols for, and you want to map those gene symbols to the Go terms or the transcript IDs. The annotation packages that are part of Bioconductor allow you to do all of these things. So you can go from a gene symbol to a gene ID, or from a gene symbol to an ID to a transcript ID. So really what we're talking about is doing any sort of mapping of any ID that you might have to another ID. And this can include functional annotation or positional annotation. So you might have a region of the gene and you want the genome, and you might want to say, what are all the genes that are in this region, or what are all the CPG sites that are in that region? So an example would be a specific goal where if you think you've got a set of data, it could be like RNA-seq or whatever, where the rows are your genes and the columns are your samples. And so you do some statistics on that. And so for every row, you've got like a t-statistic and a p-value. So then that's this block here, and you sort of tack them together so you know for each one of these genes, you've got a p-value, and maybe you're going to order that. So at the top of that, you've got the genes that are the most interesting. Well, you might want to tack onto that the gene name, the position, the gene ID, Go terms, things like that. And so the idea is to take for each one of these things and just be able to stack it together where you know all this information about the gene, and you can sort of tack it all together and have all this information together. This next little bit is about the data containers that we have in Bioconductor, and I'm going to gloss over that. Suffice it to say that there are data containers, and it makes it easy to sort of put these data together in a way that you can act as if they're a data frame, and you can subset them just like they're data frame when in fact they're not. And it's, if you're not using them, you should, is basically the take home for that. So the different annotation sources we have, we can start at the sort of lowest level here, which is, this is sort of old tech, where back when people were using microarrays, it was just all of the genes that were measured by that microarray, and it was a way to be able to map the ID from the microarray to all of the other things, the gene names and the positions and everything like that. The ORG-DBs are the next level up, where it's all of the functional annotation we have for a particular organism. So think org.hs.eg.db here is just the human entree gene-based database for everything that's known about the human. The Txdbs are the positional annotation. So this is annotation that allows us to say where genes are in the genome, where CDSs are, where transcript start sites, all that sort of information. So it's this positional information. The organism DB is a way to sort of collect the ORG-DB and the Txdb packages together, so you can get functional annotation and attach it to positional annotation easily without having to like mess around with these two different types of packages separately. The BS Genome packages are sequence packages, and it allows you to easily get chunks of the sequence of any genomic level information. So if you had say, chromosome 1, position 1 through 30, and you wanted to know what the bases are in that region, you can easily get that information from the BS genome, like this bsgenome.hsapiens HG19 thing. There's a couple others like GoDB and KegDB, where it's a way to take collections of genes that are thought to be in pathways or have the same sort of function, and it's a way of like mapping things from the gene ID to collections. And the annotation and the Biomart are online resources that we'll talk about a little bit later, but these are collections. Well, Annotation Hub is a collection of all of these type of things, and Biomart is a way to query things that are based on Ensemble IDs. So when we're interacting with Annotation DB packages, the main function that you need to use is select. If you sort of do Google searches about these things, sometimes you'll come up with these things like get or mget or totable or turning things into list. And those are sort of old ways of doing things that probably you should not use any longer. Instead, you should use the select function. And the way it works is the function name is select. You put the name of the annotation package, the things that you have that you want to map, the columns that you're trying to map to, and maybe the key type maybe the key type, sometimes you need to put that in there. So as an example, let's say we've got an AFI gene array and we've got this set of IDs right here. And we want to know what's the gene symbol for that set of IDs. So we would do, the function here would say select the DB name, these IDs, and then symbol, all caps. And it'll spit back a data frame where in that same order, we've got the probe IDs that we put as input into this select function. And then we've got the gene symbols for those genes. So if the keys that you're using are what they call a central ID, so that's the main ID for that package, you don't have to say this part up here, what's the key type. You don't have to say what the key type is. If you aren't using that key type, then obviously you do have to say. So you can always just say what the key type is. So we could have said select, and then here at the end we could have said probe ID is the key name, is the column name for this, the key type. So the question is, if you don't want to do that, how do you know what the central keys are? So if it's one of these, if it's an AFI-based or if it's a microarray-based thing, then the central keys are the probe IDs. So when I did this select hue gene, blah, 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 and these IDs are the probe IDs from that array, I didn't have to say it's the probe IDs from the array because this package is, the central ID on that package is the probe IDs. Sometimes it's in the name. So if it's org.hs.eg.db, the EG means entree gene, which is what they used to call the NCBI gene IDs. So if you have a bunch of Ensemble IDs, you could just say select org.hs.eg.db, some Ensemble IDs, and then whatever you wanted to get back, and you wouldn't have to say that it was an Ensemble ID. But you don't have to know what the central key is. You can always just say, this is the key type that I'm using. So if you need to know what the key types or columns are available for a given annotation package, you can just use this query key types, and it'll spit back. These are all the things that you can search for. So these are the things that you can ask information for. So you could ask the hue gene transcript cluster.db package for any of these key types. You could feed that, that's the input data. And then the columns are the output data. So these are all the things that you can get back out. So you can say if you've got, I don't know, uniprot IDs and you want to get out Ensemble IDs, you can do that. There is one issue with select, however. If you do something like this, where you say, we've got these 3 IDs, and then you do select, blah, 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 and we want the symbol and the map, which is the karyotype, the position in the genome. you can get one-to-many mappings. So we put in 3 IDs, and you can see here we got those 3 IDs back, but 2 through 9 is the same ID, because this is a gene that's in a lot of different places. So that can be a problem if you're trying to do this thing where you've got a row that you think is a gene, and you've got an ID, and then you want to have this one-to-one correspondence where you're making a data frame of the annotation, and then you've got your data frame of your IDs. If you have this thing where you're blowing out the rows and you've got one probe ID and then these like 8 in a row, and then this next one, then you're going to get off. So one thing you can do is you can use this function called map IDs, which gives you a control of those duplicates. So if we do the same thing, it's got the same arguments as select, and we can do the same thing where instead of saying select, we say map IDs, same arguments. The only problem is you always now have to say what the key type is. If we do that the same thing that we did here with select, we get back individual results here. So the default for map IDs is just to give you this one-to-one correspondence. And what it does by default is it just says, what's the first mapping? So here we're getting TRAF6, DDX11L1, and ARF1. And you can see that it's TRAF6, DDX11L1, and ARF1. So with map IDs by default, you'll just get one-to-one correspondence, and it's just the first one that was in that list of duplicates for each one of these that's got duplicates. So there are choices for multivals where you can say, I want to adjust how things come back. So you can say something like multivals equals a list, and then what you get back from that is a list where each list item has got all the duplicates that are in it. So you sort of will still have this one-to-one correspondence because each one of these list items could technically stack up next to a data frame. You can say character list, which is a little bit fancier version of a list where you can see it's a little bit more database-like or data frame-like where it's one-to-one correspondence. You can say filter, which does what things used to do in the past, where if you have duplicates, it just drops it, which is not super useful, but it's a thing you can do. And you can use as NA, where it just says for this one, it gives you an NA. And you can make the argument that if this thing maps to like 8 different things, then maybe we don't have that much information about it, and you just want to have an NA because you don't really know or don't trust what that thing's really measuring. So the way we usually do this, workshop is to go through something like that and then give some questions. So we've got 4 questions here that you can go ahead and try to get a little bit of practice. And I'll just give you a couple minutes if you'd like to play around with that. I'm assuming here that most people have clicked on the link that you get when you start this workshop and you've got your RStudio set up. You should have something that looks like this running. And if so, you can go ahead and do some of these things. And if you want to know what these questions are, again, you can also look in this vignettes under annotation workshop. And this is the exact thing that we're working off of. You can scroll down here and it'll have these questions.
And maybe just to point out, since if they want to play around with it, they'll need the stuff loaded in their R session, maybe show them how to run all chunks before so that they can run it.
Wait, I didn't catch that.
So in order to play around to try to answer the questions, they'll need to have the libraries and the data objects loaded in their R session. So a quick way to do that would be if you scroll down to where those chip or DB sections are and go to a code chunk, which is in the little back tick there. You could go up to the run and you can do run all previous commands.
Oh, in RStudio you can?
Yeah.
Oh, okay. Oh, I see. Run. Oh, look at that. I don't use RStudio, so I don't know these things.
While people are considering questions, it kind of was answered with the multi-evals, but we did get a question in the poll of what is the best or standard approach to deal with multi-mappings. For example, if you have Ensemble IDs and you need Entourage IDs for pathway analysis, but Ensemble IDs have multiple Entourage IDs. So is there a standard or a best way for dealing with multiple mappings?
My standard response for that is it's really difficult to do this cross-annotation mapping. So the way that NCBI decides what a gene is and where the gene lives is different than the way Ensemble does that. And so you end up with these you get these mismaps where you can't reconcile why you've got that mismatch. And so I always try to avoid doing that sort of thing where you're saying, I want to get an NCBI gene ID and I want to convert it to an Ensemble ID. If you have to do that, then it's sort of hard to know what's the best thing to do, because for each gene where you've got these multi-mapping things, you don't know why you've got that multi-map, which is why in general I tell people that they should try not to do that if at all possible. So I guess I probably won't do any of these examples because we're going to run out of time. So the Txdb packages are, again, these are positional packages, and they're used to tell you where parts of genes or entire genes live in the genome. So one of the things about the genome positioning is those are built in, so the differences between the functional annotations is functional annotations are updated like every week or every 2 weeks or every month. So the org.db.hs package is the most current functional annotations. The positional annotations, they have releases. So like this HG19 was released back like in 2005 or something like that. And so mostly this hasn't changed since. So there's a txdb.hsapiens HG19 known gene. There's one for HG38. So if you're trying to do any of this positional stuff, you need to know what build your data is based on. So in this case, if you've got HG19 data, then this is the package for you. And you can figure out what is in here. It's basically in the name. So it's a TXDB for Homo sapiens. It's based on the UCSC genome browser, the HG19 table, and their known gene mappings. We have the same thing for the Ensemble data that have basically the same naming convention. And these are, instead of being based on UCSC, which is based on NCBI data, the Ensemble is based on Ensemble data. So these usually do not necessarily agree. As with these chip and org.db packages, select and map IDs can be used to do these sort of queries. So you can say, select, here's a couple gene IDs. I want the transcript name, the chromosome, the start, the end, and I'm passing in gene IDs, and it'll spit back what you expect. So you've got gene ID 1 has got 2 positions that are slightly different. You've got these weird UCSC cryptic transcript names. And then for gene 10, you've got this other thing. Same thing is true of the Ensemble. But this isn't really normally how you would use these. Instead, you would use them to generate a G-ranges. And a G-ranges, instead of saying, I want to know something about this, I want to know something about this particular place in the genome. Instead, what you would do is you would say, just give me all the genes. So you can say genes for the Txdb hsapiens, and it'll give you this G-ranges thing that's got, this is the gene ID, this is the chromosome where it lives, this is the range in bases across that chromosome where that gene is, this is the strand it's on, and then again, it gives you the gene ID over here. And these are really useful because there's a lot of functionality that goes on behind that you can use to subset other data using a G-ranges. The one problem with just doing genes like this is some of the genes, well, there's two problems. One problem is, what is a gene? So if you've got something like a link RNA, a lot of times link RNAs will be in two places on one chromosome, and a gene is defined as the start of the first transcript to the end of the last transcript. So some of these link RNAs will start way over here on chromosome 1 and end, the second transcript will end way over here on chromosome 1. And so if you do genes, it'll be this great huge chunk of the entire chromosome 1, which obviously is not where that gene lives. The other problem with genes is you can have genes that are pseudoautosomal region where it's on the X chromosome and the Y chromosome, and something like that just gets thrown out. It doesn't, you don't see that. So if you wanted to look at something a little bit more sophisticated, you can use transcripts by. And what this will give you back is a G ranges list, where for like NCBI ID 1, it tells you there's 2 transcripts and this is where they live. It's a little bit beyond the scope of what we're doing today to get too much more into this, but suffice it to say, these are very useful and you should learn about them. And the one really nice thing about them is that they allow you to subset things based on overlapping regions. So you can do things like what genes overlap this region of the genome or things like that, which is this example right here. So I'm going to gloss over this. So for the Txdb, here's a couple of questions. One, how many transcripts does PPARG have according to UCSC? So one of the things about this is, this question is actually asking 2 things. One, what is PPARG's NCBI ID? Right, so you need to figure out what the gene ID for PPARG is using like the org.hs package. Then you need to get the transcripts and subset that G ranges list to that NCBI ID, and then you can just count basically how many transcripts it's got. And you can do the same thing with the Ensemble, like one of the Ensemble transcript IDs, transcript packages, and you'll see just for this one, if I recall correctly, UCSC says PPR gamma's got like 3 transcripts, and Ensemble says something like 26. So that's one of the reasons why I say it's hard to map between them, because Ensemble says, yeah, there's like a gajillion different transcripts for this gene, and UCSC says, no, there's like 3. And so it's hard to know, well, who's right and define what right is in that context. So at this point, Lori's going to take over, so I will stop sharing.
Before we do that, there was a poll question of how to convert HG19 gene symbols to corresponding gene symbols in HG18, ensuring one gene mapping.
Well, so for gene symbols, so the only thing that's got a sort of build thing to it, so like HG18 and HG19 only refer to where we think those things are in the genome. The gene symbol never change. Well, the gene symbol can change, but it doesn't change based on where that gene is in the genome. So like PPAR gamma, they might say, oh, you know what, we thought it was on chromosome 1 right here. And then later on they say, oh, you know what, we're wrong, it's on chromosome 3 over here. But it's always going to be called PPAR gamma. So the name doesn't actually change. So the org.hs.eg.db package only gives you those sort of functional, only gives you that sort of functional information, and it's sort of orthogonal to where they are in the genome.
Sorry, James, to interrupt. My question was regarding the aliases that keeps on, like the gene names get updated.
Aliases?
Yeah.
Well, so there is, right. So there is in, let me go to here. So one of the things you can do, I'm hoping you can see this, is I think I have to load this. Let's say we want to know all the aliases for whatever one is, whatever gene ID one is. So aliases, as they go through time, people have called this particular gene multiple different things. And the HUGO consortium says this is the current gene symbol, which in this case I think is first one there. So right, so A1BG is the symbol for Entree gene ID #1. But in the past, people have called it ABG and GAB and HIST 2477 and A1BG. So sometimes you can have like data that you got that was older, and they're still calling it GAB. So then you can have this issue where you need to say, well, they used to call it GAB and they're not calling it that now, but I need to map it to get the Entree gene ID to get like where it is in HD38 or whatever. Then you have to use, so you could do something like, right? So if you think it's an alias, you can do it like that, where you say, I have this ID GAB, I want the symbol, and I'm pretty sure it's an alias. Does that make sense?
Thanks, James. That answers my question.
Okay, great.
And I do see a number of questions racking up in the polls. I'm going to present just a little bit longer, and then maybe I'll end a little early, so hopefully we can come back to some of these and do some more demonstrations. We did want to show a few more things. So as James just ended with, you could see that there was some cross between the orgDBs and the Txdbs. So naturally, the next question is, if you wanted to use information from both of these objects, is there an easier way to do it? And there is, to some extent, in Bioconductor. So the first are these organism DB packages, which are like these meta packages that contain all of the information from an orgDB, a Txdb, and a GoDB, all together in one database object that you can use your standard, the standard functions that you saw in orgDB, like genes or transcripts by, the same ones that you saw in Txdb for creating ranges, and you could use all of them to grab and utilize all of the information together. And everything is there. The downside to the organism DBs is that if we look at how many there are in Bioconductor, so this is the main Bioconductor website here. If we go to the list of packages, and we're talking annotation packages here, so this is the list of all of the annotation packages available in Bioconductor. And I'm going to go to package type, and I'm going to go to organism DB, and unfortunately there's only 3. So unless you're working with the main Homo sapiens mouse or rat, then you wouldn't be able to use these orgDB metadata packages. But they are available if you are doing human, rat, or mouse for use. If you're using some of the less well-known organisms, there is the package organism dplyr. And this should make some of you tidy people happy that we're going to be introducing a little of the dplyr methods in tidyverse where you can pipe along the way. And the organism dplyr is nice because, as with the organism DB packages, you're still going to get all of the functions available from the orgDB or the Txdb packages. So your key types, your selects, your exons, your promoters, your gene functions, those will all still work on your organism dplyr object. The main function in organism dplyr is this src_organism function. And we did provide in organism dplyr, there is this HG38 Lite, which is a condensed kind of example database to be used for workshops, so we could load that. And then as a proof of principle, once we get that SRC object, you can use the promoters function that we saw from the Txdb object. And I guess backtracking a little bit, you can see when we look at this SRC that there's a bunch of different tables. So we have ID, we have Go terms, we have the ranges that we would see from the Txdb objects. So all of those are tables in this SQLite database that we created. And to access one of those tables, there's a table extractor. So we could see all of the information provided by each of the different tables. So we can grab the table ID and see that that table contains the map and Ensemble ID, gene symbol, a gene name, and any sort of alias. So that might be a useful table depending on what you were looking for. And organism dplyr, through the dplyr methods, provides a way to make these complex queries between all the tables underlying the database. So for example, say we wanted to get the start and stop locations for particular gene symbols, you could join those two tables together, filter by any gene of interest. In this case, we filtered by the symbol names ADA and NAT2. And then maybe we wanted to change the order and we wanted to look at the chromosome and start and end position, the strand, the symbol, and if it had any sort of alias. So if we put those into the select, then we get our table, our joined table, and cross information for everything that you would normally get from the two separate ORGDB or Txdb. And we do have some organism dplyr methods. I will mention in Bioconductor, and I'm not sure how many of you are familiar with it, or have gone over it, or really new to Bioconductor and to our packages, but for any package that you load, you can get help information for functions of a package by doing a $sign. So if you do a $sign and a function name for a package, it will bring up how you use the function and maybe some other useful information about what the arguments of the function are expecting. So you can see here that maybe if you wanted to know what organisms are supported using organism dplyr, there's a supported organism function to tell you what would be available. And you could explore what organisms could actually be utilized through organism dplyr. Genome packages are another type of annotation packages, and this would be a type of package that you would get sequence information from. And there is the main Genome package, and you can list the available genomes available. And the main function of the Genome package is a get sequence function. So once you load a Genome package, you could get a sequence for an entire chromosome. You could get a sequence for any sort of subrange by passing a GRange object. James touched on what a GRange object was. So if you had particular genes of interest, you could create a GRange list or a GRange object to pass into the get sequence, and then you could get specific sequences for those maybe transcripts or exome sequences of interest. And there are some Genome exercises there too. So I guess before we get into the online resources, are there any quick questions that might be able to answer? Okay, so a brief overview of the Annotation Hub. So an Annotation Hub is, it allows you to download annotation resources and query annotation resources from different annotation objects without having to install them. So for the Annotation Hub, we can, I'll do this live here. It's a collection of resources that are contributed from the community. So anyone can submit resources to the Annotation Hub for distribution to the community at large. And currently in, well, I guess we have to download it first. Give it a second to load. So the current snapshot is from July 20th of 2020. And if we look at our Annotation Hub object, there are 50,282 different types of records and resources that are available for use that you could download at any time. And there are a variety of different annotation resources. And the best way to do it is to query. And querying can take a little bit of, I think we put in here, it's kind of like a well-formed Google search. And you can search across any of the metadata columns of the hub, like title or data provider, source type, and our data class. So if you perhaps had a data provider that you were looking for of interest, UCSC maybe, or Ensemble, you could search the Annotation Hub resources that are currently available for those specific data providers. Another useful one is our data class. So if you're used to working with GRanges objects, or you're used to working with a 2-bit file, you could search for those particular R data objects so that you had those to work with. You could also subset by species or by the source type. So when you form these queries, you can query against the hub to find out what resources are available, and then download the resources by calling a specific Annotation Hub resource of interest. And I want to make sure that we have time to get some of your questions answered. I am part of the core team, and we do have the core team lunch talks on Thursday and Friday. So if anyone has specific questions about the hubs, feel free to bring them up there just so that we have time to get to some of the poll questions here. So I'll unshare, and then maybe James can pop back up too, and maybe we could tackle some of the poll questions.
So I guess the top poll question is, does the Annotation Hub hold old versions of the functional annotations corresponding to the org packages from previous releases?
So it does, but because the org DBs are tied to specific releases and specific versions of Bioconductor, you wouldn't necessarily always have access to them. So the org DBs in the Annotation Hub that are provided would be whatever is appropriate for the current release and version of R. So are there ways to interact with annotations like these in less common model organisms like bacterial species?
Yes, depends on what, right, so there's different ways. One, you can use the Annotation Hub and you can do a query, a well-crafted query, something like org.orgdb, and if you wanted the functional annotations, orgdb, and then something like the species and probably genus and species, and then you'll be able to see if there's an annotation package in the Annotation Hub. You can also, if the data are on, if it's not in the Annotation Hub, there's a package, there's a function in Annotation Forge that you can use to make a Txdb package from NCBI. And so that will automatically go to NCBI and download all of the data and then parse out just the species that you're interested in. That's all dependent on there being enough data at NCBI for that to be useful. Sometimes with the less common bacterial species, you end up going through downloading a metric ton of data and parsing it to find out that NCBI knows about like 3 genes. So you can do it, but sometimes it's a lot of work and then you find out at the end that it wasn't worth it. The advantages and disadvantages of using Biomart instead of the DB packages. The main advantage of using Biomart is it's based on Ensemble IDs. So if your IDs that you have in hand are Ensemble IDs, I would recommend using Biomart, or if you're looking for positional data, one of the Ensemble DB packages rather than anything based on NCBI, just because then you don't have that issue with trying to map between the IDs. So for sure, Biomart's way better than the .db packages if you have Ensemble IDs, or basically if you have Ensemble IDs, it's much better to do that. So there's another question: why are some genes in a Txdb very large? That's something that I talked about earlier, where the definition of a gene, at least in a Txdb, is where does the start of the first transcript start and where is the end of the last transcript. And if you do genes for the Txdb, some of those are these link RNAs, which are really usually pretty short, and you might have one little short link RNA way upstream and then one way downstream. And by definition, genes will give you the start of that first one all the way to the end of the last one. And it'll say, yeah, this link RNA is 90% of the length of the chromosome, which obviously is not right. But that's just an error based on the simplification of transcripts to genes. I mean, genes don't really exist, transcripts exist. So a gene is just a convenient way of describing a group of transcripts. Oh, there's a good question for you, Lori. Are the data immutable in the Annotation Hub?
So yes, so the data in Annotation Hub should be fairly stable. We try to add a new entry if any of the data is updated, so that you could always backtrack and reproduce your research with the actual original Annotation Hub object that you downloaded. So it doesn't forget. So if you wanted to use an updated object, or if someone made some sort of update to the annotation instances, it would generally have a new Annotation Hub ID. We are working on a versioning in the Annotation Hub, so it would be a little bit easier to find the different versions, but right now it would have a completely different Annotation Hub ID if it was updated.
Would the existing one still be there? So
Yeah, the existing one would still be there too.
Right, so if there's an Ensembl or something that I used, I don't know, 2 releases ago, and I remember that AHID and I just punch that back in, I'll get that same one.
If it was contributed by an outside user, as long as they didn't request it to be deprecated, it would still be there, yes. Or if they haven't requested it to be removed, then yes, it would still be there.
What's the best way to deal with one-to-many mapping in the case of Ensemble IDs or gene names? That's a good question. I don't really have an answer for it. Yeah, I don't really have an answer for it. I mean, so you can have these instances where you are doing something like, I have a gene ID and it maps to like 3 different ensemble IDs or something like that. It's hard to know what's the best. The sort of the naive thing to do is just to use map IDs in the default, because then it'll just take the first one. And I don't know that that's any better or worse than doing anything else. I mean, it's sort of hard to take one-to-many mappings and know without programmatically to do that. I mean, if you went and looked at every gene and said, oh, why does this have the one-to-many mapping? And you sort of research it and figure out why, then you can come up with a plausible answer why you think that, this is the actual good mapping. But programmatically, if you have 20,000 genes you're trying to annotate and 3,000 of them have a one-to-many mapping, I don't know what you do about that other than say, well, I'll just take the first one, or I'll take a random one, or whatever.
I'd say I see the best way to create a database for your species. I would still say using Biomart and Annotation Forge is probably the way to go. I don't know if you have any opinions of that, James, but as far as I'm aware of, those are kind of the standard ways to create ones for your own purpose and own use.
Yeah. Yeah, if it doesn't exist.
When would you use an Ensemble DB as opposed to an org DB? I was wondering if Johannes might be on the call.
Yeah, the choice for me between an Ensemble DB and a Txdb is basically, so in that case, you're really talking about, well, you did some RNA-seq or some single-cell RNA-seq, right? And so you've done this mapping where you said, I've got these reads that map to the genome or map to these transcripts. And then you say, well, what are those transcript IDs? So if you used Ensemble IDs, let's say you got the GenCode transcript file and you use that to do the mapping, then all of your IDs are going to be Ensemble IDs. In that case, I'd use an Ensdb package just because then you're not doing this cross mapping. If you went to NCBI and got a FASTA file there of all the transcripts and you use that to decide, or if you used a Txdb to do your, you've got your bunch of BAM files and you did summarize by overlaps and you get all the counts for all the genes and it's based on Ensemble, on Entrez gene IDs, then I'd use a Txdb just because then you don't have to, there's none of this cross mapping that I find so problematic. To get intronic regions. There's an introns, right? There's an introns function, I believe, right? So I just did introns by transcript, and this is what it spits back. So for like Entrez gene 1, here's the 2 introns that exist for that one. Here's the introns for Entrez gene 2. So that's how you would get them. What do you think about the MyGene package? Do you know anything about the MyGene package, Laurie?
I do not. So I guess I have no opinion except that I have to look into it.
I have no idea. What's the fastest way of getting the total exonic length for each gene for RPKM normalization? I think exons by plus reduce plus width plus sum. I don't know about this question. Is it possible to predict annotated gene ranges belongs to a hyper-hypomethylated regions in a chromosome? So if I took this question to mean I have these genomic regions where I know that it's hypermethylated within this region, then you can convert that to a G ranges where you have, here's all my regions, it's on all these chromosomes, and here's the chromosomal regions. Then you can easily just do gene or transcripts by from a Txdb and then just use subset by overlaps. So you can subset the gene transcripts by the hypo or hypermethylated region, so long as you have the G ranges for it. So the poll questions went away?
I was managing them while we were answering
Oh,
so that
you're
we
making
could see new ones. Yeah.
Okay.
So did anyone have any, we have about 4 minutes left to wrap up here. I will say that normally we give you guys time to work through those exercises that are in the vignette. And as far as I'm aware, these will, the vignettes and the pathable will stay around. I will upload a file I have as a little cheat sheet of answers, but we encourage you to kind of work through them on your own. And if you have questions or want to understand better or don't understand how something works, we'll post the answers so that you have them available, but please feel free. I'm sure James will agree to reach out to either of us if you want more explanations on anything.
So James, I would like to ask something about the introns by transcript function. So supposedly I want first intronic regions and separately and the rest of the intronic regions separately. So can that be achieved using this function, or we have to do it?
Can you restate? I don't know if I followed.
Sorry?
I don't know if I understand your question. Can you restate it?
So like we want to separate the first intronic region from rest of the intronic regions.
Oh, okay. There's probably a really smart way of doing that using, because it'll be a list. That might be a Martin question. Martin, do you know how to do that really easy? Where you just want to take and you've got a GRanges list and you just want the first range from every.
Yeah, it's you could try using head 1. So it's like a list, and head applied to some list of these list objects. Head sort of acts on the individual elements and not on the list itself. But I'd actually have to explore that a little bit.
So it's just a head function?
Maybe.
Yeah, usually when people ask questions like that, I come up with some convoluted thing, and then
Yeah.
or Michael Lawrence comes along and they're like, no, that's like one function, dude.
It's a great question to ask on the support site, and then you can watch James reply, and then, as he says, Hervé come on and come up with a very clever.
Yes. Here's my convoluted way of doing that stupidly. Now Hervé or Michael Lawrence will come along and be like, no, that's not it.
Because this question was repeatedly asked on BioStars, and like there isn't a convincing solution to it, so that's why I asked it.
So you said head, right?
Yeah.
So I will mention that we're at time. So if anyone we do have 10 minutes. So anyone that wants to stay over can. But if you were going to try to make the next workshop, you probably should sign off and sign on to the other.
No, head just does the first one, Martin.
Head will probably operate on the capital L list rather than the GRanges inside each list. So you would have to L apply over the or endo apply over the list and then head that.
That's going to be super slow though, isn't it?
I don't know.
Usually L apply over GRanges is like brutally slow.
You could do something with lengths and unlist and relist.
Yeah, I mean, usually you
Choose
want
the first element of the.
right.
I'm just smiling at this challenge, which.
Thank you all for coming.
Thanks. If you have any questions, you know where we are.
Okay, James, we can like follow up later on this. I'll also try at my end.
Okay.
Thanks a lot. This was a wonderful conversation.